A carbohydrate. Carbohydrate is a biomolecule consisting of carbon C, hydrogen H, and oxygen O atoms, usually with a hydrogen-oxygen atom ratio of, as in water, and thus with the empirical formula CmH2ON, where M may be different from N. This formula holds true for monosaccharides. Some exceptions exist. For example, deoxyribose, a sugar component of DNA, has the empirical formula C5H10O4. The carbohydrates are technically hydrates of carbon. Structurally, it is more accurate to view them as aldoses and ketoses. The term is most common in biochemistry, where it is a synonym of saccharide, a group that includes sugars, starch, and cellulose. The saccharides are divided into 4 chemical groups: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides and disaccharides, the smallest, lower molecular weight carbohydrates, are commonly referred to as sugars. The word saccharide comes from the Greek word
sákkharon,
saccharon, meaning sugar. While the scientific nomenclature of carbohydrates is complex, the names of the monosaccharides and disaccharides very often end in the suffix -ose, as in the monosaccharides fructose, fruit sugar, and glucose, starch sugar, and the disaccharides sucrose, cane or beet sugar, and lactose, milk sugar. Carbohydrates perform numerous roles in living organisms. Polysaccharides serve for the storage of energy, e.g. starch and glycogen, and as structural components, e.g. cellulose in plants and chitin in arthropods. The carbon monosaccharide ribose is an important component of coenzymes, e.g. ATP, FAD and NAD, and the backbone of the genetic molecule known as RNA. The related deoxyribose is a component of DNA. Saccharides and their derivatives include many other important biomolecules that play key roles in the immune system, fertilization, preventing pathogenesis, blood clotting, and development. They are found in a wide variety of natural and processed foods. Starch is a polysaccharide. It is abundant in cereals, wheat, maize, rice, potatoes, and processed food based on cereal flour, such as bread, pizza or pasta. Sugars appear in human diet mainly as table sugar, sucrose, extracted from sugarcane or sugar beets, lactose, abundant in milk, glucose and fructose, both of which occur naturally in honey, many fruits, and some vegetables. Table sugar, milk, or honey are often added to drinks and many prepared foods such as jam, biscuits and cakes. Cellulose, a polysaccharide found in the cell walls of all plants, is one of the main components of insoluble dietary fiber. Although it is not digestible, insoluble dietary fiber helps to maintain a healthy digestive system by easing defecation. Other polysaccharides contained in dietary fiber include resistant starch and inulin, which feed some bacteria in the microbiota of the large intestine, and are metabolized by these bacteria to yield short-chain fatty acids. Contents terminology. In scientific literature, the term carbohydrate has many synonyms, like sugar in the broad sense, saccharide, ose, glucide, hydrate of carbon, or polyhydroxy compounds with aldehyde or ketone. Some of these terms, especially carbohydrate and sugar, are also used with other meanings. In food science and in many informal contexts, the term carbohydrate often means any food that is particularly rich in the complex carbohydrate starch, such as cereals, bread and pasta, or simple carbohydrates, such as sugar, found in candy, jams, and desserts. Often in lists of nutritional information, such as the USDA National Nutrient Database, the term carbohydrate or carbohydrate by difference is used for everything other than water, protein, fat, ash, and ethanol. This includes chemical compounds such as acetic or lactic acid, which are not normally considered carbohydrates. It also includes dietary fiber, which is a carbohydrate but which does not contribute much in the way of food energy, kilocalories, even though it is often included in the calculation of total food energy just as though it were a sugar. In the strict sense, sugar is applied for sweet, soluble carbohydrates, many of which are used in food. Structure. Formerly the name carbohydrate was used in chemistry for any compound with the formula CmH2ON. Following this definition, some chemists considered formaldehyde, CH2O, to be the simplest carbohydrate, while others claimed that title for glycolaldehyde. Today, the term is generally understood in the biochemistry sense, which excludes compounds with only one or two carbons and includes many biological carbohydrates which deviate from this formula. For example, while the above representative formulas would seem to capture the commonly known carbohydrates, ubiquitous and abundant carbohydrates often deviate from this. For example, carbohydrates often display chemical groups such as NaCETYL, e.g. chetan, sulfate, e.g. glycosaminoglycans, carboxylic acid, e.g. sialic acid, and deoxy modifications, e.g. fructose and sialic acid. Natural saccharides are generally built of simple carbohydrates called monosaccharides with general formula CH2O and wherein is 3 or more. A typical monosaccharide has the structure H, CHOH X, C equals O, CHOH YH, that is, an aldehyde or ketone with many hydroxyl groups added, usually one on each carbon atom that is not part of the aldehyde or ketone functional group. Examples of monosaccharides are glucose, fructose, and glyceraldehydes. However, some biological substances commonly called monosaccharides do not conform to this formula, e.g. uronic acids and deoxy sugars such as fructose, and there are many chemicals that do conform to this formula but are not considered to be monosaccharides, e.g. formaldehyde CH2O and inositol, CH2O. The open-chain form of a monosaccharide often coexists with a closed ring form where the aldehyde ketone carbonyl group carbon, C equals O, and hydroxyl group, O, react forming a hemiacetal with a new COC bridge. Monosaccharides can be linked together into what are called polysaccharides, or oligosaccharides, in a large variety of ways. Many carbohydrates contain one or more modified monosaccharide units that have had one or more groups replaced or removed. For example, deoxyribose, a component of DNA, is a modified version of ribose. Chetan is composed of repeating units of NaCETYL glucosamine, a nitrogen-containing form of glucose. Division. Carbohydrates are polyhydroxyaldehydes, ketones, alcohols, acids, their simple derivatives and their polymers having linkages of the acetal type. They may be classified according to their degree of polymerization, and may be divided initially into 3 principal groups, namely sugars, oligosaccharides and polysaccharides. The major dietary carbohydrates class, DP*, subgroup, components. Sugars. Monosaccharides, glucose, galactose, fructose, xylose. Disaccharides, sucrose, lactose, maltose, trehalose. Polyols, sorbitol, mannitol. Oligosaccharides. Maltooligosaccharides, maltodextrins. Other oligosaccharides. Raffinose, stachyose, fructooligosaccharides. Polysaccharides, greater than, starch, amylose, amylopectin, modified starches. Non-starch polysaccharides, glycogen, cellulose, hemicellulose, pectins, hydrocolloids. DP* equals degree of polymerization. Monosaccharides. Main article, monosaccharide. D-glucose is an aldehexose with the formula C, H2O. The red atoms highlight the aldehyde group and the blue atoms highlight the asymmetric center furthest from the aldehyde. Because this O is on the right of the Fischer projection, this is a D-sugar. Monosaccharides are the simplest carbohydrates in that they cannot be hydrolyzed to smaller carbohydrates. They are aldehydes or ketones with 2 or more hydroxyl groups. The general chemical formula of an unmodified monosaccharide is H2O, literally a carbohydrate. Monosaccharides are important fuel molecules as well as building blocks for nucleic acids. The smallest monosaccharides, for which N equals, are dihydroxyhistone and D and LGLYCERALDEHYDES. Classification of monosaccharides. The
μικρό α,
and
μικρό β,
Anomers of glucose. Note the position of the hydroxyl group, red or green, on the anomeric carbon relative to the CH2OH group bound to carbon. They either have identical absolute configurations R or S,
Alpha,
or opposite absolute configurations R, S or S, R.
Β, 15.
Monosaccharides are classified according to three different characteristics: the placement of its carbonyl group, the number of carbon atoms it contains, and its chiral handedness. If the carbonyl group is an aldehyde, the monosaccharide is an aldose. If the carbonyl group is a ketone, the monosaccharide is a ketose. Monosaccharides with 3 carbon atoms are called trioses, those with 4 are called tetroses, 5 are called pentoses, 6 are hexoses, and so on. These two systems of classification are often combined. For example, glucose is an aldehexose, a 6-carbon aldehyde, ribose is an aldopentose, a 5-carbon aldehyde, and fructose is a ketohexose, a 6-carbon ketone. Each carbon atom bearing a hydroxyl group O, with the exception of the first and last carbons, are asymmetric, making them stereocenters with 2 possible configurations each, R or S. Because of this asymmetry, a number of isomers may exist for any given monosaccharide formula. Using Le Belvan-T Hoff rule, the aldehexose D-glucose, for example, has the formula C(H2O), of which 4 of its 6 carbon atoms are stereogenic, making D-glucose one of equals possible stereoisomers. In the case of glyceraldehydes and aldotriose, there is one pair of possible stereoisomers, which are enantiomers and epimers. Dihydroxyacetone, the ketose corresponding to the aldose glyceraldehydes, is a symmetric molecule with no stereocenters. The assignment of D or L is made according to the orientation of the asymmetric carbon furthest from the carbonyl group. In a standard Fischer projection, if the hydroxyl group is on the right, the molecule is a D sugar, otherwise it is an L sugar. The D and L prefixes should not be confused with D or L, which indicate the direction that the sugar rotates plane polarized light. This usage of D and L is no longer followed in carbohydrate chemistry. Ring straight chain isomerism. Glucose can exist in both a straight chain and ring form. The aldehyde or ketone group of a straight chain monosaccharide will react reversibly with a hydroxyl group on a different carbon atom to form a hemiacetal or hemictyl, forming a heterocyclic ring with an oxygen bridge between 2 carbon atoms. Rings with 5 and 6 atoms are called furanose and pyranose forms, respectively, and exist in equilibrium with the straight chain form. During the conversion from straight chain form to the cyclic form, the carbon atom containing the carbonyl oxygen, called the anomeric carbon, becomes a stereogenic center with 2 possible configurations. The oxygen atom may take a position either above or below the plane of the ring. The resulting possible pair of stereoisomers is called anomers. In the
Μικρό αλφα,
anomer, the O substituent on the anomeric carbon rests on the opposite side trans of the ring from the CH2OH side branch. The alternative form, in which the CH2OH substituent and the anomeric hydroxyl are on the same side cis of the plane of the ring, is called the
μικρό β,
anomer. Use in living organisms. Monosaccharides are the major source of fuel for metabolism, being used both as an energy source, glucose being the most important in nature and in biosynthesis. When monosaccharides are not immediately needed by many cells, they are often converted to more space-efficient forms, often polysaccharides. In many animals, including humans, the storage form is glycogen, especially in liver and muscle cells. In plants, starch is used for the same purpose. The most abundant carbohydrate, cellulose, is a structural component of the cell wall of plants and many forms of algae. Ribose is a component of RNA. Deoxyribose is a component of DNA. Lactose is a component of lysoflavin found in the human heart. Ribulose and xylulose occur in the pentose phosphate pathway. Galactose, a component of milk sugar lactose, is found in galactolipids in plant cell membranes and in glycoproteins in many tissues. Mannose occurs in human metabolism, especially in the glycosylation of certain proteins. Fructose, or fruit sugar, is found in many plants and in humans. It is metabolized in the liver, absorbed directly into the intestines during digestion, and found in semen. Cellulose, a major sugar of insects, is rapidly hydrolyzed into 2 glucose molecules to support continuous flight. Disaccharides. Sucrose, also known as table sugar, is a common disaccharide. It is composed of 2 monosaccharides, D-glucose left and D-fructose right. Main article. Disaccharide to join monosaccharides are called a disaccharide, and these are the simplest polysaccharides. Examples include sucrose and lactose. They are composed of 2 monosaccharide units bound together by a covalent bond known as a glycosidic linkage formed via a dehydration reaction, resulting in the loss of a hydrogen atom from one monosaccharide and a hydroxyl group from the other. The formula of unmodified disaccharides is C12H22O11. Although there are numerous kinds of disaccharides, a handful of disaccharides are particularly notable. Sucrose, pictured to the right, is the most abundant disaccharide, and the main form in which carbohydrates are transported in plants. It is composed of 1 D-glucose molecule and 1 D-fructose molecule. The systematic name for sucrose,
alpha,
DGLUCOPYRANOSYO, rightwards arrow, DFRUCTOFURANOSIDE, indicates 4 things. Its monosaccharides, glucose and fructose, their ring types. Glucose is a pyranose and fructose is a furanose. How they are linked together, the oxygen on carbon number, C1 of
alpha.
D-glucose is linked to the C2 of D-fructose. The -side suffix indicates that the anomeric carbon of both monosaccharides participates in the glycosidic bond. Lactose, a disaccharide composed of 1 D-galactose molecule and 1 D-glucose molecule, occurs naturally in mammalian milk. The systematic name for lactose is
Vita,
DGALACTOPYRANOSYO, rightwards arrow, DGLUCOPYRANOSE. Other notable disaccharides include maltose, 2 D-glucoses linked,
alpha 1,4,
and cellulose, 2 D-glucoses linked.
Vita 1,4.
Disaccharides can be classified into 2 types, reducing and non-reducing disaccharides. If the functional group is present in bonding with another sugar unit, it is called a reducing disaccharide or biose. Nutrition. Learn more. This article may lend undue weight to certain ideas, incidents, or controversies. Grain products. Rich sources of carbohydrates. Carbohydrate consumed in food yields 3.87 kilocalories of energy per gram for simple sugars, and 3.57 to 4.12 kilocalories per gram for complex carbohydrate in most other foods. Relatively high levels of carbohydrate are associated with processed foods or refined foods made from plants, including sweets, cookies and candy, table sugar, honey, soft drinks, breads and crackers, jams and fruit products, pastas and breakfast cereals. Lower amounts of carbohydrate are usually associated with unrefined foods, including beans, tubers, rice, and unrefined fruit. Animal-based foods generally have the lowest carbohydrate levels, although milk does contain a high proportion of lactose. Organisms typically cannot metabolize all types of carbohydrate to yield energy. Glucose is a nearly universal and accessible source of energy. Many organisms also have the ability to metabolize other monosaccharides and disaccharides, but glucose is often metabolized first. In Escherichia coli, for example, the lactoperon will express enzymes for the digestion of lactose when it is present, but if both lactose and glucose are present, the lactoperon is repressed, resulting in the glucose being used first. See deoxy. Polysaccharides are also common sources of energy. Many organisms can easily break down starches into glucose. Most organisms, however, cannot metabolize cellulose or other polysaccharides like chitin and arabinoxylans. These carbohydrate types can be metabolized by some bacteria and protists. Ruminants and termites, for example, use microorganisms to process cellulose. Even though these complex carbohydrates are not very digestible, they represent an important dietary element for humans, called dietary fiber. Fiber enhances digestion, among other benefits. Based on the effects on risk of heart disease and obesity in otherwise healthy middle-aged adults, the Institute of Medicine recommends that American and Canadian adults get between % of dietary energy from whole grain carbohydrates. The Food and Agriculture Organization and World Health Organization jointly recommend that national dietary guidelines set a goal of % of total energy from carbohydrates, but only % directly from sugars, their term for simple carbohydrates. A 2017 Cochrane systematic review concluded that there was insufficient evidence to support the claim that whole grain diets can affect cardiovascular disease. Classification. Nutritionists often refer to carbohydrates as either simple or complex. However, the exact distinction between these groups can be ambiguous. The term complex carbohydrate was first used in the U.S. Senate Select Committee on Nutrition and Human Needs publication Dietary Goals for the United States, 1977, where it was intended to distinguish sugars from other carbohydrates, which were perceived to be nutritionally superior. However, the report put fruit, vegetables and whole grains in the complex carbohydrate column, despite the fact that these may contain sugars as well as polysaccharides. This confusion persists as today some nutritionists use the term complex carbohydrate to refer to any sort of digestible saccharide present in a whole food, where fiber, vitamins and minerals are also found, as opposed to processed carbohydrates, which provide energy but few other nutrients. The standard usage, however, is to classify carbohydrates chemically, simple if they are sugars, monosaccharides and disaccharides, and complex if they are polysaccharides, or oligosaccharides. In any case, the simple versus complex chemical distinction has little value for determining the nutritional quality of carbohydrates. Some simple carbohydrates, e.g. fructose, raise blood glucose slowly, while some complex carbohydrates, starches, especially if processed, raise blood sugar rapidly. The speed of digestion is determined by a variety of factors including which other nutrients are consumed with the carbohydrate, how the food is prepared, individual differences in metabolism, and the chemistry of the carbohydrate. The USDA's Dietary Guidelines for Americans 2010 call for moderate to high carbohydrate consumption from a balanced diet that includes 6 1-ounce servings of grain foods each day, at least half from whole grain sources and the rest from enriched. The Glycemic Index (GI) and glycemic load concepts have been developed to characterize food behavior during human digestion. They rank carbohydrate-rich foods based on the rapidity and magnitude of their effect on blood glucose levels. Glycemic index is a measure of how quickly food glucose is absorbed, while glycemic load is a measure of the total absorbable glucose in foods. The insulin index is a similar, more recent classification method that ranks foods based on their effects on blood insulin levels, which are caused by glucose, or starch, and some amino acids in food. Health effects of dietary carbohydrate restriction. Main article, Low carbohydrate diet. Low carbohydrate diets are associated with increased mortality, and they can miss out on the health benefits afforded by high-quality carbohydrates such as is found in legumes including grain legumes or pulses, and fruit and vegetables. Disadvantages of the diet might include halitosis, headache and constipation, and in general the potential adverse effects of carbohydrate-restricted diets are under-researched, particularly for more serious possible risks such as for bone health and cancer incidence. Carbohydrate-restricted diets can be as effective, or marginally more effective, than low-fat diets in helping achieve weight loss in the short term. The Endocrine Society state that, when calorie intake is held constant, body fat accumulation does not appear to be affected by even very pronounced changes in the amount of fat versus carbohydrate in the diet. In the long term, effective weight maintenance depends on calorie restriction, not the ratio of macronutrients in a diet. The hypothesis proposed by diet advocates that carbohydrate causes undue fat accumulation via the medium of insulin, and that low-carbohydrate diets have a metabolic advantage, has been falsified by experiment. It is not clear how low-carbohydrate dieting affects cardiovascular health. Any benefit from HDL cholesterol might be offset by raised LDL cholesterol, which risks causing clogged arteries in the long term. Carbohydrate-restricted diets are no more effective than a conventional healthy diet in preventing the onset of type diabetes, but for people with type diabetes they are a viable option for losing weight or helping with glycemic control. There is little evidence that low-carbohydrate dieting is helpful in managing type diabetes. The American Diabetes Association recommends that people with diabetes should adopt a generally healthy diet, rather than a diet focused on carbohydrate or other macronutrients. An extreme form of low-carbohydrate diet, the ketogenic diet, is established as a medical diet for treating epilepsy. Through celebrity endorsement, it has become a popular weight loss fad diet, but there is no evidence of any distinctive benefit for this purpose, and it risks causing a number of side effects. The British Dietetic Association named it one of the top worst celeb diets to avoid in 2018. Metabolism. Main article: Carbohydrate metabolism. Carbohydrate metabolism denotes the various biochemical processes responsible for the formation, breakdown and interconversion of carbohydrates in living organisms. The most important carbohydrate is glucose, a simple sugar monosaccharide that is metabolized by nearly all known organisms. Glucose and other carbohydrates are part of a wide variety of metabolic pathways across species. Plants synthesize carbohydrates from carbon dioxide and water by photosynthesis, storing the absorbed energy internally, often in the form of starch or lipids. Plant components are consumed by animals and fungi, and used as fuel for cellular respiration. Oxidation of 1 gram of carbohydrate yields approximately kcal of energy, while the oxidation of 1 gram of lipids yields about kcal. The human body stores between 300 and 500 g of carbohydrates depending on body weight, with the skeletal muscle contributing to a large portion of the storage. Energy obtained from metabolism, e.g., oxidation of glucose, is usually stored temporarily within cells in the form of ATP. Organisms capable of anaerobic and aerobic respiration metabolize glucose and oxygen aerobic to release energy, with carbon dioxide and water as byproducts. Catabolism. Catabolism is the metabolic reaction which cells undergo to break down larger molecules, extracting energy. There are 2 major metabolic pathways of monosaccharide catabolism: glycolysis and the citric acid cycle. In glycolysis, oligo and polysaccharides are cleaved first to smaller monosaccharides by enzymes called glycoside hydrolases. The monosaccharide units can then enter into monosaccharide catabolism. ATP investment is required in the early steps of glycolysis to phosphorylate glucose to glucose, phosphate, G6P, and fructose, phosphate, F6P, to fructose 1,6-biphosphate, FBP, thereby pushing the reaction forward irreversibly. In some cases, as with humans, not all carbohydrate types are usable as the digestive and metabolic enzymes necessary are not present. Carbohydrate chemistry. Carbohydrate chemistry is a large and economically important branch of organic chemistry. Some of the main organic reactions that involve carbohydrates are: carbohydrate acetylation cyanohydrin reaction,
Lobrie de Bruin van Ekenstein transformation,
Amadori rearrangement Neff reaction, Wald degradation, Koenigsegg reaction, carbohydrate digestion.